actually have access to the who, I mean, it was made for people actually doing this, but who else? Is there any idea that at universities, for instance, people were even looking at these things?
Well, that's a very interesting question, the subject of from lived experience to the written word, actually, which is the incredible burgeoning of interest in practical knowledge in the 16th century. Just the huge interest in recipes, recipe manual or recipe books, collections of recipes were went through many editions, were kind of bestsellers, some of them. And, nobles went into artisans' workshops, artists' workshops. There was great conversation about, in commissioning works of art by all kinds of people at this time. So the interest in art as practical knowledge, as power, as either the representation of power, the theater of state, or as the kind of work that goes into canon casting, for example, was important to state power at this time.
Because this could be in future years, when we go on talking about that, you may be aware or not be aware how important recipes are for our scientists here.
Oh, yeah.
They all read this, and then how they use the text and how they actually generate their experiment may vary. Some experiments fail and others not. So this process of
Protocols,
how they use protocols and what they are made for is interesting.
Yeah.
Should add on to what Herman just asked. Do you have evidence of when manuscript recipe books became printed manuscripts? Is there any correlation?
Oh, no, I mean, it's, completely interpenetrated, manuscript and printed text. I mean, there are some examples that you can trace way back into manuscripts, but of individual recipes, but this manuscript, what was it made for? We don't actually know. It's anonymous, etc. But also it is probably was the basis of a publishing project that never materialized.
I think we have to go on.
Yes.
Because the next, I think is going to see patient options.
Yeah, I think we're now.
No,
So it's my immense pleasure to introduce next speaker.
Roy, can we get rid of this?
Hello.
I think it's
Hello. Yeah. Okay. It's a great pleasure to introduce next speaker following Pamela's wonderful talk. And actually, I couldn't help but notice that in one of your slides, the last slide where she introduced, where you introduced some of these publications from Regeneration, Thomas Lozito, who is our next speaker, you have cited his paper as well. So Tom has been doing some really, really wonderful work on lizard tail regeneration. As you probably know, lizards are very famous for living behind that tail when there is a predatory circumstances, and after a while they regrow this tail. Now, I particularly work with salamanders, axolotl, which are considered champions of regeneration. They regenerate tissue flawlessly. And although lizard can regenerate, there are some flaws. And Tom's work over last decade basically has highlighted why those flaws exist in lizard tail regeneration and what are the molecular mechanisms behind it. So let's welcome Tom. Tom did his PhD at University of Cambridge and NIH. And then right now he's at University of Southern California as an assistant professor in the Department of Orthopaedic Surgery, Regenerative Medicine, and Stem Cell Biology. Thank you, Tom.
Thank you so much, Brian. Can everyone hear me? Is this okay?
You could.
All right, great. Well, thank you so much to the organizers for this unique opportunity to talk about my research in this interesting context. As it was mentioned, I'm fascinated by lizards. So why would I devote my life to lizards? Well, lizards are amazing for a number of reasons. Many of these are due to their intermediate evolutionary position between the amphibians and the mammals. And this intermediate position affects much of their biology, including their regenerative biology. So lizards are the only amniotes and closest relatives of the mammals that retain adult regenerative capabilities that you see in the amphibian salamanders. So developmentally, genetically, and in terms of their cell types and tissue types, adult lizards are very similar to adult mice, but somehow lizards retain their abilities to regrow amputated tails. Now, because of their, because of their intermediate position, they have this affects much of their regenerative capabilities. So while the amphibian salamanders can regenerate both limbs and tails on the one side, and on the other side, you have the mammals that really can't regenerate any appendages as adults. The lizard is only able to regenerate its tail, not its limbs. So focusing on the tail, here I'm going to present lizard and salamander and mouse tails 0 to 4 weeks following amputation. And these dashed lines mark the amputation planes. So again, while the mouse tails scar over, lizards and salamanders spontaneously regenerate new tails. However, if you focus in on the lizard tail, you can start to notice something strange about the regenerated lizard tail. So you can always tell a lizard with a regenerated tail because the lizard is unable to match the color and the scalation of the original tail. This makes lizards unique in that they regenerate an imperfect regenerate, and these imperfections are more than just skin deep. So here I'd like to show micro CT X-ray images of axolotl salamander, lizard, and mouse tails now 10 weeks following amputation. And while the salamander tail achieves the same skeletal patterning as the original, the regenerated lizard tail skeleton is unsegmented and unpatterned, and just takes the form of this unsegmented cartilage tube. So these imperfections extend to all the tissues of the regenerated lizard tail. The muscle is disorganized and unsegmented. The blood vessels of the regenerated tail lack valves. The spinal cord lacks new neurons. There's no true tendons being formed in the regenerated lizard tail. But again, the most obvious of these imperfections concerns the skeleton. And that's because the regenerated lizard tail skeleton is almost completely cartilaginous. The regenerated lizard tail skeleton takes the vertebrae of the original tail, but it's regenerated as a single unsegmented cartilage tube. Now, the vast majority of this lizard tail tube resists ossification and remains as cartilage for the lifetime of the lizard. And it's actually this unique cartilage tube that first attracted my attention to lizards. Cartilage is a tissue that mammals, and humans in particular, are almost completely unable to repair. And here's an animal that spontaneously regenerates an abundance of cartilage spontaneously that resists ossification for the lifetime of the lizard. And I really wanted to know how they're able to make this cartilage. And we began by characterizing the developments of the lizard cartilage tube. And here I'll describe some key developmental stages because they motivated many of the studies that I'll describe in this talk. For each of these stages, I'll show some very simple cartoons above representative sagittal sections of tails analyzed by pentachrome staining, which stains bone in red, cartilage in green, muscle in kind of this purplish red, and spinal cord tissue again in this purple. And again, these dashed lines denote the amputation plane. So immediately after tail amputation or tail loss, the tail stump contains a terminal vertebra and of course the severed spinal cord through the middle. By about 7 to 10 days, wound epidermis migrates over the stump and thickens to what's called an apical cap. At the same time, immune cells recruited to this injury site begin secreting proteases, which degrade tissues, and cells liberated from these degraded tissues begin to accumulate under the apical cap. These liberated stump cells proliferate, forming a mass of mesenchymal cells called the blastema, which is a very important regenerative structure The blastema gives rise to a majority of the regenerated tail tissues. During this blastema stage, the spinal cord regenerates as an ependymal tube and invades the blastema. And this kind of trifecta of blastema cells, regenerated spinal cord, and wound epithelium kind of drives the rest of the tail regeneration process and then differentiation of the blastema cells. So over about a month, the cartilage tube forms around the regenerated spinal cord, and the cartilage tube makes contact with the original tail vertebra at its proximal end. You also get regenerated fat as well as regenerated muscle. So this histological image kind of shows the simplified version, the simplified structure of the regenerated lizard tail, basically tubes within tubes. We have the spinal cord ependymal tube in the middle, the cartilage tube surrounding that, encased by basically a tube of fat, and then a tube of muscle, and finally the skin. So to investigate this process and these questions that we have, I decided to use a very special lizard, the mourning gecko. This is an all-female species that exhibits parthenogenesis or asexual clonal reproduction, where the mom lizards spontaneously lay fertilized eggs. This is the only lizard species that is both diploid and capable of tail regeneration and reproduces in this manner. I picked this lizard species because all of the offspring are genetically identical. We have a colony of over 1,000 of these mourning geckos, and they derive from a single female that I brought to the lab about 10 years ago. So we take advantage of this genetically identical species because we can take cells and tissues and transplant them amongst any member of our colony without fear of rejection and without having to use immunosuppressive drugs, which we have found to interfere with the regenerative process. So we use this lizard species in our background in regeneration to ask the following questions. We want to know what are the relevant cell types and molecules responsible for driving tail regeneration. And ultimately, we hypothesize that copying these cells and signals can improve wound healing in an appendage or a species that doesn't naturally regenerate. So the first simple experiments I want to describe are asking the roles of two of those tissues that I talked about, the spinal cord and the wound epithelium, in driving tail regeneration. So we basically performed experiments where we surgically removed these tissue types. So again, intact lizard tails with intact epithelium and spinal cords regenerate normally with a blastema, followed by regenerated spinal cord and cartilage tube. Removal of a spinal cord from the very distal stump of the amputated tail completely interferes with regeneration. You don't get regeneration at all. In fact, you get a scar formation very similar to what we see in amputated mouse tails. Similarly, removal of the epithelium around the stump tissues interferes with the wound epithelium formation. And again, that totally ablates the tail regeneration process. So because we're using the mourning gecko, we can start to take advantage of that by doing some rescue experiments. So here we took recipient lizard tails that have had their spinal cord removed, and then we attempted to try to rescue tail regeneration by applying either wound epithelium, grafts, or regenerated spinal cord implants taken from donor lizards implanted in these recipient lizard tails to see if we can rescue tail regeneration. And again, a lizard tail with everything intact regenerates normally. Spinal cord removal inhibits tail regeneration. Now, when we apply a wound epithelium graft to these tails lacking spinal cord, we can actually recapitulate the early stages of tail regeneration. We get stump cell liberation and some proliferation and accumulation underneath the wound epithelium graft, but this process stalls and we don't get potentiation of the blastema formation or further tail regeneration. However, amazingly and very interestingly, we could totally rescue tail regeneration merely with the implantation of spinal cord implant. This spinal cord implant doesn't even have to be implanted in the location of the original spinal cord. Really anywhere in the tail stump will result in more or less normal blastema formation followed by tail regeneration. So just by applying a replacement of the spinal cord tissue, you're able to rescue tail regeneration. We wanted to test the boundaries of these findings, and we started to apply these wound epithelium and spinal cord implants on lizards that didn't even have an amputated tail, that didn't have an amputation injury. And what we found was that just by applying these tissues really anywhere along the lizard body, we can induce ectopic tissue growth. So anywhere on the lizard that we applied the wound epithelium graft would result in a kind of an ectopic blastema formation with an accumulation of cells underneath the wound epithelium graft. But again, this wouldn't potentiate into a full-on tail. However, we could basically grow an ectopic tail anywhere on the lizard simply with the subcutaneous implantation of a regenerative spinal cord implant. Would grow these tail-like structures along the body of the lizard, and inside these tail-like structures, it has a normal-looking cartilage tube, regenerated muscle, all within these structures. And doing some fluorescent microscopy here, looking specifically for collagen 2, a marker for cartilage in green, EDU, a marker for cell proliferation in red, and MHC, a marker for regenerated muscle in this whitish color. You can see just by applying these different tissues, you're actually able to get new different tissue formation in these regenerative structures. And in particular, just with the application of a regenerated spinal cord implant, a tissue that has really nothing to do with cartilage, that's able to induce this new cartilage tube being formed in the regenerative structure. So of course, in the very first couple slides, I mentioned lizards are unique in that they're really the only animal within the animal kingdom that incorporates a non-regenerative structure, a limb, with a regenerative structure, their tail, in the same animal. So this actually affords us some interesting opportunities for investigation because now we can take cells and tissues that we collect from the regenerative tail and apply them to the naturally non-regenerative limb to see if we can spark regeneration in an appendage that really never evolved to regenerate. And as you might guess, we see the very similar things that I've just described in the last few slides. A wound epithelium graft to apply to an amputated lizard limb, first of all, prevents the scar formation that forms under control conditions. And you actually see some new tissue liberation and also new tissue formation underneath this wound epithelium graft. However, again, the most interesting case was the application of these regenerated spinal cord implants, which basically resulted in the growth of these tail-like extensions from the limb. So these are to us, very exciting and interesting results. These spinal cord implants, even in the limb environment, they don't regenerate a new limb. Again, they're restricted to formation of tail tissues, but it was definitely an improvement over the control case. Again, we inhibited scar formation. We had new blood vessels, new muscle, and new cartilage being formed just with the application of the spinal cord implants. So again, these conclusions from this first set of studies is that the spinal cord is imperative to lizard tail regeneration. We can use these spinal cord implants to induce a tail-like limb regeneration in lizards. And we went on to study the specific cells within these spinal cord implants that were responsible for these biological activities. We've identified a population of neurostem cells within the spinal cord tissue that's responsible for many of these tail regeneration-inducing capabilities. Future work, of course, we're aiming to characterize the expression profiles of these subsets of spinal cord neurostem cells towards identifying the specific factors responsible for their regenerative capabilities. If there's still time, I'd like to talk about another project that we have in the lab here, focusing on different lizard species. Just check the clock. Yeah. Okay, so
Yes, you can go on for some time.
okay, thank you. Okay, so here we use different lizard species with different regenerative potentials. So now the first lizards that evolved from amphibians, maybe 750 million years ago, these through the fossil record have been shown to have tail regenerative capabilities. And through the diversification amongst the various lizard groups, this ability to regenerate tails was lost in some of these lizard groups. Some of these losses occurred relatively early in these different lizard speciations, whereas some of them have occurred relatively recently, within the last million years or so. So this project is going to focus on one of these more recent losses, the Coralophus geckos from the island of New Caledonia. This is kind of a real life Jurassic Park. There's no native mammals, kind of reptiles run the show. And there's some giant geckos that have diversified on this island that show some differences in tail regenerative capabilities. So for example, there's two gecko species in this genus Coralophus, the crested gecko and the Sericinorum gecko. And the Sericinorum gecko can regenerate its tails, but the crested geckos cannot. However, this is one of the few instances in nature where we have two species with divergent regenerative capabilities that are close enough in relation that they can hybridize. And my lab is one of the first, the only lab really that I'm aware of that has hybridized these lizards and studied their F1 hybrids to see what effects mixing these two lizard species had on regenerative capabilities. And we found that the F1 hybrids of these two animals, in fact, could regenerate their tails. So interestingly, these hybrids exhibit kind of more or less intermediate appearance and biology between the two parents. You can see the crested gecko, so named because it has these crests along both its head and these eyelashes over its eye, whereas the Sericinorum gecko lacks any of these ornamentations. The hybrids lack the head crests, but still have the eyelashes. And then if you look at the tails of these animals, the crested gecko has a short patterned tail, whereas the Sericinorum gecko sort of has a long, unpatterned tail, where the hybrids have kind of long patterned tails. So again, intermediate between the two species. And of course, when you talk about their regenerative capabilities, the Sericinorum gecko is able to regenerate a full tail. The crested gecko, the regeneration fails, whereas the hybrid is able to regenerate a tail, albeit slower than its regenerative parent. So again, tracking the tail growth over time, we see that the three species exhibit similar early phases of tail elongation. However, the crested gecko plateaus at just a couple millimeters, and additional growth, no additional growth is achieved. However, the Sericinorum gecko takes off after this initial growth phase, and the hybrid species also takes off after this plateau, but not as fast as the Sericinorum parent. So we profiled differences in the marker expression between these two lizard species, these two lizard groups, and found that day 14 blastema from all three lizard species expressed CD59 in their distal regions, but only the Sericinorum gecko and the hybrid expressed the marker SOLF1 in their distal regions. So we hypothesized that the Sericinorum gecko and its hybrid achieve a distal blastema population that is required for tail regeneration, something that the crested gecko can't achieve, can't form for some reason, and thus unable to regenerate further. So we've shown this in a number of ways, but one of the coolest ways I wanted to show is with these cell transplantation studies. We found that the cells of both the parent species are able to be transplanted in the tails of the hybrids without rejection. So it's something by hybridizing these species, you basically create an environment that supports the survival of either parent cells, which I think is really cool. And these transplanted cells are incorporated into the regenerated tail blastemas. So we labeled crested gecko and Cerasnorm gecko with different color fluorescent dyes, green dye-O for the crested gecko cells and a red dye-I for the Cerasnorm gecko cells, and planted them into the hybrid tail stumps and tracked where the different colors were localized to following amputation. And the dye-O labeled crested gecko cells localized in proximal locations where the dye-I labeled Cerasnorm cells were evenly distributed and the only cells found in the distal regions. So again, we think the differences between the non-regenerative and regenerative Coralophus geckos is the ability of these blastema cells to make this distal cell identity. And we're investigating using this model to investigate this further. I believe my 20 minutes is up. I, of course, have other projects that I could talk about, or I could stop, for discussion.
Yeah, we can move on to discussion, Tom.
Okay. Then I'd just like to end by acknowledging my lab and our funding sources, and I look forward to any questions or, of course, discussion on these topics.
Thank you, Tom. There is a question in the audience.
How did you come up with your?
So my life has been a little transformed. It's wonderful to introduce Rebecca.